Good afternoon, everyone. Thank you, Geoffroy and Juliette, for here,
No,
I think, for the invitation. So it's a pleasure to be here and to present the work I've been doing during the past few years, and also the one that I'm doing now. So as Geoffroy said, I'm going to talk about amorphous calcium carbonate precursors, organic molecules, and how together they are able to form colorful single crystalline biominerals. So before I start, I would like to acknowledge my collaborators. So the work I'm going to show today are mainly published in these papers, and it has been gathered during my postdoctoral stay, as Geoffroy said, at the Max Planck Institute of Colloidal Interfaces, led by Peter Fratzel, and under the supervision of the committee and in close collaboration with the committee. We both are moved to TU Dresden in Germany as well. Zhao Yongzhu was very much involved in that work as well. He is now a professor at the University, and Professor Zolot Toyako from Technion University in Israel, as well as Professor Gilbert from University of Milton, were also involved. And since I moved back to Paris 4 years ago, so I'm working in close collaboration with Nadine Nassif and Thierry Azaïs from the LCMCP lab as well, and with Frédéric Marin from the University of Bordeaux. So what do I mean by colorful biominerals? So these are these biominerals that are formed by sea urchins in particular. So we work with the Paracentrotus libidus species, which is populated French coast, and you can appreciate the variety of colors even within one species. We're in particular interested by the spines, and if you look at the cross-section of sea urchin spines, you can see that the colors is also very homogeneously distributed within the spines, and even in one spine you have some color variation. So these biominerals are convoluted porous structure, and we know that by thanks to X-ray computed tomography that you can see as well in the lab. And if you look at one virtual section along the longitudinal axis of the spine, you can appreciate the porosity. So you have some dense parts here called the septa, which are responsible for some bending stress properties. But if you look at the center here on the cross section, you can see this porous 3D porous structure, which is a 3D mesh of thin trabecula, which is called the steron. And it's actually the structure that you can find in all echinoderms. And it has micro lightweight properties. That's why the sea urchin test has been performed so well. And they are single crystalline. Although they have this very complex morphology, they diffract X-ray as single crystal of calcite. They are enriched with magnesium. And when you record XRD pattern at the synchrotron, for example, here was at BC, and you do some rotation of your single crystal along the C-axis of calcite, you sum up all the diffraction pattern, and you obtain this typical pattern from single crystalline materials with a bright spot. But if you have a closer look and on only one pattern, you can see that you have some diffuse, what we call diffuse scattering around the bright spot. So it's not a perfect single crystal, and it is actually a mosaic crystal. And what is a mosaic crystal? It is when you observe some small changes in the orientation of some micrometer size crystallites, which despite their slightly differences in orientation are largely coherent. And so one question that has been in the field for many years is why these convoluted biominerals diffract as single crystals. And this is where biomineralization comes in. And so what was the question is how come in the lab we are able to synthesize calcite crystal with completely classical morphology, and organisms are able to build biogenic calcite which have amazing shapes. I'm bringing here only a few examples in the echinoderm one, but you have many different examples all over. So one concept which has been brought is like so the presence of amorphous calcium carbonate precursor phase could help in building such biominerals. The role of as well of intra-crystalline organic molecules, how they can shape the different form of crystals as well as organic templates. And today I will focus on the two first concepts. So if we have a look at amorphous calcium carbonate in biology, so these are just a few examples that I bring in here. two categories have been identified. So the so-called stabilized ACC category, where you can find some amorphous calcium carbonate in the form of different animals. So for example, acidian speakers in crayfish gastroliths, as well in the gastroliths of the lobster and in the cuticle, and in some plants, for example, cystoliths in the ficus. And the second category, okay, so what I should say here is like in the stabilized ACC, they are all hydrated. And most of the time they are in presence of inorganic ions like magnesium or phosphate or some organic substances. And the second category is transient ACC. And these are ACCs that are temporarily stabilized, but then eventually they will convert into more stabilized, into crystalline polymorphs, which is thermodynamically more stabilized than ACC. And it has been first evidenced in the sea urchin larva speaker, and later on some corals as well. So what I should mention here is like all these ACC are extracellular ACC formed by eukaryotes, and so I'm not mentioning your beautiful bacteria with the cyanobacteria. So in particular, this cyanobacteria with the amorphous calcium carbonate particles. So here we really focus on what is extracellular ACC formed by eukaryotes. And so one question which has been raised is how come this metastable ACC phase does it exist at all and it's stabilized in these organisms? And many research groups have tried to answer this question by synthesizing in the lab amorphous calcium nanoparticles in the presence of inorganic ions, for example. And this is what we did a few years ago already. So Xiaoyong synthesized a series of ACC with different amounts of water in the presence of magnesium or phosphate. We are trying to have some low content to be relevant to biology. And what we could show, and what was already known, is like these ions indeed stabilize ACC against crystallization, either if the crystallization is induced by heating during the SC measurement, or induced by high humidity content in isothermal calorimetric measurement. And by doing a bit of data analysis, you can integrate the area under the peak, extract the enthalpy. By considering the entropy as well, you extract the fluid energy, which tells you about the thermodynamic or kinetic stability of your system. And it turned out that all these ACCs that we synthesized were actually kinetically stabilized and not thermodynamically as it could be thought at first. So we wanted to understand a bit better what was the mechanisms of stabilization by looking at the local structure. So asking the question, are those ACCs, which have obviously different stabilities, have also different local structure? So some work has been done previously on biogenic ACC. So you can recognize the ACC we'll talk about further before. And some XLAPS measurements were done, and they could show that the short range order, at least, of this ACC were different among different animals. However, so with XLAP, we are only element specific, so we measure at the calcium edge, and we have access up to 3 angstrom. And we knew by per distribution function analysis from total X-ray scattering that actually ACC has as well medium range order. So it has an order up to 8-10 angstrom. So we were wondering if we could use this technique to also probe and measure this range and see if we could have some different structure in our different ACC. So briefly, to have a small overview, if you're not familiar with the technique, so what we do, so we have our sample, we shot it by X-rays, and we have the typical the diffusion pattern of an amorphous phase. But what is important for PDF analysis is to have a very long range, a very long Q range. And once you integrate your signal, so this would be actually the range you measure with a normal diffractometer D8, for example, in the lab. But thanks to this setup, you have access to all this Q range as well. And this is where we are interested for making the PDF. Because after making some correction, you can actually enhance what is happening here. And by doing a Fourier transform, then you get your PDF. And this is a typical PDF of an amorphous calcium carbonate phase, and we have indeed an order up to 8 angstrom. So we did this for all our sample, and we were a bit disappointed because we couldn't see any difference difference in the PDF. So basically, this is so in the real space, and we have access to distance between ion pair. But under one peak, you can have some average. So you have at the same time calcium-oxygen and oxygen-oxygen distances. So we were not able to see any difference in the structure, but we tried hard. And to do that, we induce the crystallization. So we induce the crystallization here. I'm showing the results for the heating. And we could calculate a PDF for every temperature and try to see if we could see differences during the rearrangement. And we did see some difference, but in order to enhance these differences, so we calculated what we call the differential dPdFdt. So we basically subtract one PDF to the other at each temperature, and then we see the difference. So what you see here is like the crystallization form at 160, and we do see some structural rearrangement here, going into the direction of solid state transformation rather than dissolution or precipitation. And if you do the same with your different samples, so in the presence of magnesium, we could evidence some difference, some slight difference in the structural rearrangement, indicating that these inorganic ions are actually bound to the water, and which decreases the mobility of the system, and that's why somehow they stabilize the ACC against crystallization. So let's move to the ACC in real life, let's see. So in organisms. So again, we look at URCHIN, the URCHIN spine. So here's the URCHIN, a growing spine. Can you pay attention to the change of color here? It would be important for later. So the growing spine here, very fresh growing spine. So it's mainly pink, and as it grows and converts fully to calcite, it becomes. And it was first evidenced by Alberic in 2004 that we had some nanoparticles in the spine, which were diffracting electron as amorphous. And it was proposed that the crystallization was occurring via secondary nucleation phase, meaning like one particle will crystallize and will trigger the crystallization of the other, and etc. And it was also known that, in particular, at the larva stage, so a lot of work has been done at the larva stage actually. And so first ACC was evident in the larva, and 10 years later, it was proposed that we had the presence of hydrated ACC, which converted into anhydrous ACC and finally into calcite. So this was shown by doing calcium examination with X-ray photoemission electron microscopy, but not only. I will come back to that later. But we were asking, is it the same in the spine? So here you have the short level, section level stages, because not much was known about biomineralization of the spine apart the work from Yale. And so we were asking this question. And so we went to ALS, as well to the CIT to do some X-ray photoemission electron microscopy. And we could obtain this spectra, the calcium LH. And basically, the hypothesis is like, according to the intensity of the crystal field splitting peaks, you could differentiate between if your calcium carbonate is crystalline or if it's amorphous, and even if it's hydrated or anhydrous. So this was what we obtained on the biogenic sample. But that was not known before. We had, in this time, we were able to have some synthetic reference. And for our synthetic reference, we had a similar symmetry. These are spectra which has been averaged. And just for you to have an idea of how it looks like, so this is also average, but then with a huge background when you measure the data. So you have to be careful about the interpretation of these peaks we were discussing a week ago, but still now. But still, we are pretty confident by comparing with our reference that we do have these three phases. And we could identify them in the spine as well, and suggesting that we had similar sequence at the larva stage and at the adult stage. So to continue the story and the formation of calcite in spine, so now we look at the mature spines. And it was shown 10 years ago that in the mature spines, when in principle all this process has been done, that we still have some remaining ACC in the structure. which was stable, but we wanted to go a bit further, and in order to determine if this ACC was anhydrous, hydrated, how was the stabilization mechanism, how come it was still in the structure after the biomineralization was finished. What was the role of ACC? Eventually, together with organics molecule on lattice distortion, calcite lattice distortion, because it's known in many biominerals that the presence of organics changes the calcite structure, and in particular decreases the lattice parameter. So what did we get? So we perform TGA together with DSC, and again we identify the crystallization peak, and by integrating we identify 8% of ACC. And because we had almost no water loss, we could show that it was anhydrous. And if you remember from before, all the stabilized ACC evidence in biology were all hydrated. But this one was anhydrous with 0.1 to 1 ratio of water calcium. So we propose that this ACC was actually kinetically trapped during the crystallization of this hydrated phase into the anhydrous. So some of the anhydrous phase would convert in magnesium calcite, whereas some ACC was still kept in the structure. Okay, so about the role of organics. So this is a pinna prism, calcitic pinna prism, and which shows in many of the different minerals about this lattice distortion. So how we measure lattice distortion in calcite? So again, we need quite high resolution XRD data, and this you obtain unfortunately only at the synchrotron, because you need to be able to have very good results, so very thin half width of half maximum, to be able to perform good read-but refinement and extract the lattice parameter. And once you extract the lattice parameter, you can compare to geological calcite, which has no organics. And this is what you see here. So to start at room temperature, we have 0.15 percentage, meaning that what I said before was the T-axis is a bit higher when you have some organics, but when you start to burn the organic, then the lattice relax, and you go close to a geological calcite. But in this systems, we didn't have ACC. And now we wanted to check in our system with ACC how it would look like. So we did the same. We heated the sample, we crystallized the remnant ACC, we burned the organic at 400 degrees, and we measured the lattice distortion. So as for the other biominerals, we had a decrease of the lattice parameters, but this was pretty new. Like the increase during ACC crystallization was something new. So the fact that we had the decrease when organic was burning is indicating that we have a strong inorganic interaction. And this increase in particular will mean like actually the strain we have in calcite in all biominerals is built up when we have this transformation of ACC to calcite. Because when you crystallize, then the organics are all around and compensates the change in volume that you have during ACC crystallization, because the density is not the same, so you have a decrease. And we could conclude that in our system we had ACC layer adjacent to the inorganic-organic interaction. I'm not sure how much time I have. Okay. So in order to go further in this heat-mediated transformation, which helped us to understand how the system is built, we did some SAXS measurement the same on 15 samples, and we could identify like the appearance of this streak, which is actually an interface which is creating, and which is indicating on how the organics was before being burned. And if you integrate your signal, so by doing, I don't want to go into the details, but this bump here, which is moving as you hit the samples, it's the presence of some nano inclusion that will draw while you burn, you transform your LCC and you burn your organs. So putting all this data together, we could propose a very simplistic model of how the structure was. So with the presence of organics in the form of nano-inclusion surrounded by LCC layer, and with some organics as well perpendicular as well as along the CAXS. So what we could propose is like ICC crystallization, so proceed as far as the organics can serve as space filler and accommodate for density changes. And beyond that point, so crystallization is hindered and ACC kinetically trapped in the structure. So in the time I have left, I'd like to share with you what we are doing now in the lab since 4 years. And we are looking at the biomineralization together with pigmentation. So I already present the variety of color we can observe in parasympathetic spine and this sign here. So far we just look at the biominerals if it was a dead tissue, basically, so without all the tissue or all the cells, without just the mineral. But in real life, it's covered of tissue, so epidermis, dermis, which is full of cells, and in particular, there is this very interesting cells, which is the red spheroid cells, which are producing the pigment, which is a polyhydroxylated concomitant molecules. So there is about 41 different molecules, but the one we are particularly interested in are those ones. So you have spinal from A, B, C, E, equivalent from A. So these are naptokinone molecules with some hydroxyl a little bit everywhere. And so what we are asking in the group now is like we're trying to understand the origin of the color variation among the spines and during biomineralization. So is it due to pH? So we call this pigment pH-NQ. Is it due to a different pH-NQ composition or different interaction it might have with other organics or ions? And how the pH-NQ molecules get incorporated within the gluon spines, because they are really inside the mineral. When you bleach with sodium hypochlorite the spine, the color stays. And the last question we are asking is do these pH-NQs have a role in biomineralization mechanisms as well? So it can have some play on ACC stability, as we saw before for inorganic ions. If they influence crystal growth by the morphology of the structure of the crystal, etc. So I'm going to focus on the two last part, which is the work of 2 PhD students in the group, which are just behind here. So first is Claudio. So briefly, Claudio, so is developing some extraction protocol from the spine and from the red spheroid cells. So from the spine, it's a collaboration with Frédéric Marin from the University of Sorbonne. And briefly, it's a 2 weeks process, so it's quite long, and we obtain very, very little. But still, we managed to analyze the final part. So what we do, we dissolve. We need to get rid of all the calcium carbonates. So we try to do this as little as possible. We get different fractions. And the last fraction, which is mainly composed of pigment, we then need to get rid of all the salt. So we do some ether extraction, some purification, and we manage to get one sample. And in the case of purple spine, it's mainly this molecule, which is spinal from A. And for the protocol is developing in collaboration as well with the PE station in Plaintia in Spain. So it's extracted from the cell, it's extracting the celomic frill of the sea urchin, which is like your blood basically, and you have many different kinds of cells. So it's like fractioning the cell and is obtaining pure population of red spheroid cells and is extracting. And the interesting thing is that you compare both molecules are different. So in one case, in the case of the cells where the pigments are produced, it's mainly one molecule, which is a keynote from A. Whereas the molecule which is ending in the spines is a different one. This molecule is also produced here, but in very little quantity. So it means we have a selective incorporation of this pH-NQ within the spine. So this was one of his first results. And what we also observed is like previously, thanks to FIBSEM images of auto-freeze substituted samples. I don't know if you're familiar with this technique, but you high pressure freeze your sample, and then you progressively replace all the water by acetone, you dry it, and then by resin. So it's quite invasive, and you stain it with potassium. But still, you manage to get some nice image, and we could really catch the moment where the pigments here are getting out from the cells, and this is a lot of hypothesis, but this is the basis of our work, and where the pigments are incorporated into the spine. And the beauty of FIPSEM is like you can reconstruct in 3D, and we have an idea of how the cell looks like all around close to this mineral, which is but it's just, as I said, the basis of our work. So we want to get closer to that. And what Claudio is doing, we start like we step back, and we really try to characterize the cells and the vesicles in the cell. So we still need to verify this maybe by the EDS measurement or stem yields, but from the morphology, at least, this looks like pigments. But so what has been done so far, and TM and these samples have been done a long time ago, but people were not able to maintain the samples, and all the pigments were lost. So now Claudio will optimize the protocol with the SAXS from at the museum, and they managed to conserve the pigment, and they even managed to identify this sugar layer all along one vesicles. That for us, it's indicative that these pigment molecules and the environment here must be acidic. It has been shown in archaea, apparently, that indeed the sugar layer here might protect the cell from any products that might be toxic or nocive to the cell. So these are first insights about the role of this pigment in the cell, even before getting into the mineral. And the last project I would like to share with you is the work of Bhaskar, who is also here, a third-year PhD. And here, so we go back to the lab and to make some bio-inspired synthesis of calcium carbonate and in the presence of this pigment. As you could understand from before, it's quite tedious protocol to extract this pigment. So at the moment, we work with commercially available naphthoquinone, and they are not so complex as the one in URCHIN, but still they do the job. So they have 2OH instead of fuel. And as a powder, it looks very red. And so it plays with concentration, and he introduces in a sodium carbonate term. And the nice thing about these pigments in general is like they are pH sensitive, so they change of color with pH. And we work here at pH 11.5, so we are in the blue stage. And as soon as it's precipitating calcium carbonate by calcium chloride, either in the form of ACC, if you take it at 10 seconds, or in the form of calcite, if you let it run for 48 hours. And these are the samples, the powder sample we got, so they are quite blue according to the concentration, which is different from this red we started with. So we measure the pH while the synthesis is running. That's why we know, like at 10 seconds, we still have ACC. So by measuring the pH, we know like this plateau is is because we have ACC, and as soon as the pH drop, we have the crystallization first into laterite and calcite, and then we can pull in calcite if we wait 48 hours. And by doing this experiment, what you can see is like the addition of naphtazarine is delaying the crystallization of calcite and laterite in water. So we kind of have a stabilization as well of the naphtazarine molecule in the crystallization. But we wanted to know if this where is this pigment? Is it only on the surface? Is it inside? Is it just absorbed? And we run for Bhaskar a very fast experiment with bleaching. So with sodium hydrochloride 1%, 2 hours, and we could see, well, first for calcite, like the color was not changing. So the pigments were really incorporated within the structure. Whereas for the case of ACC, it's completely white. You can't even see that, meaning like the pigment was destroyed or removed. being weakly bound to the SAC, whereas in the case of calcite, it's really occluded with it. And you may wonder how it affects the morphology. So this is the typical ACC nanoparticles. You can synthesize in the lab in the presence of the naphthalene at different concentration. Doesn't affect so much the size of the ACC. It was known before that adding phosphate, for example, would reduce the size of your particles, but the addition of pigment does not. And for the crystal morphology, so this is the control, we do have an effect on the surface. So you can see the surface becomes very porous, and yeah, it tends to be even quadratic pores. So we do have an effect on the naphthalene molecule on the morphology of the surface. So, and as before, so you might remember this curve. So now in the presence of naphthalene, we could also, could also show that it was stabilizing the ACC. But again, with PDF, we couldn't see any difference. But this time, even by trying hard, during the in-situ heating, we couldn't also see any difference. So it looks like all the ACC have the same structure, local structure up to medium range order, and the stabilization comes from somewhere else, like at larger scale. And finally, we looked at the calcite crystal and the lattice distortion. And here again, in the presence of naphthalene molecule, we could not evidence any shift of the peak, even by heating, meaning like we didn't have lattice distortion. It was shown for amino acids, for example, at the same concentration, we would be able to have lattice distortion up to 0.005, but for us was really significant. Meaning that the naphthalene molecule are incorporated into the calcite, but is not interacting into the lattice. So which is quite new compared to all other molecules that has been tried so far. So we're a bit puzzled, but we are looking still on this data. And maybe one last input, because we are in the lab or in a team actually that does a lot of solid state NMR. I know that some of you collaborate with Thierry as well. And a solid state NMR is a very good tool to probe the inorganic-organic interaction with such systems. So this is why we tried at first. So this is how solid state NMR of naphthalene pure looks like. So it's a carbon spectra. And you have only 3 peaks, meaning like you have 3 equivalent carbon due to the symmetry of your molecules and the exchange that could happen between this hydrogen and oxygen. So in different color here, you have this 3 carbon. Okay, now when you introduce it in calcite, so you have the carbonate peak of the calcite, but all the peak related to naphthalene shifts to lower chemical shifts, and this shift is quite high, meaning that your molecule is different somehow. And interestingly, this shift, even if the signal is really low, but it's the same for ACC and calcite. So now we are wondering how this molecule looks like, because it's different for sure. So we know like it can be deprotonated, so either mono or deprotonated according to the pKa. We work at this pH, and we thought because of the color range, it might be fully deprotonated. But theoretical studies showed that these are the pKa, meaning like we should be actually in this stage, but we still need to finish this study and complete. So yeah, so with that, I'd like to conclude briefly. So I hope I show you a bit how URCHIN looks like. So they have strained calcite in biominerals, and it's mostly due to these volume changes between ACC to calcite during the crystallization, and compensated by the organics which are all around. So it's really like ACC and organics together, which at the end makes the properties, at least at the structural point of view, of the biogenic calcite. And so a lot of work needs to be done on pH into molecular pigmentation and biomineralization, but I think it's going well. And there is a lot of potential on these molecules. And what I didn't say as well is like they've been studied for many years, but in a different field, in pharmaceutical field, because they have antioxidant and many different kind of properties, and are in the immune system of the CH. So So we really think like they have an impact as well on biomineralization. And even some people might argue that they are here because they were there and they got entrapped by chance, but they might be not so much by chance. And yeah, so I'd like to acknowledge our collaborators, the amazing people at the different line we've been to, the different students that have been joining the team so far and did the work. And recently, Asher Schmidt, who was a visiting professor in our lab, helped us a lot for interpreting the NMR results. And I thank you very much for all for your attention, and I look forward to the questions. Thank you.
Yeah, thank you very much, Marie. Does anyone has a question currently? Yeah.
Well, I didn't ask about the visualization last time we met, but I still have a lot of questions, but two main questions I want to ask. One is about the organics part. How much do you think is amount of organics needed in order to see these effects in reduction, the stabilization, the kinetically trapped ACC, which is in the transit state? And the second question is more of like, maybe you mentioned this, but so you did mention that you were not able to see differences in the PDF of different ACC. Mm-hmm. But with the variation of the amount of organics, did you also don't see any changes in the short range structures of those ACC, or that also is similar as to PDF? Like, is there no short range differences, short range structure differences of ACC with different varying amount of inorganic additives or organic additives, or is it the same like as you see for the PDFs? So those are.
Okay, so first one is quick. So you meant for the synthetic system, right? So VESICLES is varying the concentration between 0.3 and 1.5, well, 0.15 to 1.5 molar percent of organics. So quite little. We're trying to fit what is happening in vivo. So in vivo, we have about 1% weight percent of organics. And of all together, because it's not only pigments, it's also proteins and polysaccharide and of pigments. We have much lower, like 0.1, let's say maximum weight percent. So we try to work at low concentration, but this is what has been also used for amino acids, for example, and with the same concentration, and still they could show some lattice dysfunction. Okay, and for the second question, so I think maybe it was not so clear. So for PDF, you don't look at medium range order only. You look from short range to medium, actually from short range to long range order, if we would have to measure in long range, but it turned out that for ACC, we just have to go to medium range. And so also in the long range, also in the short range order, we don't see differences. Maybe you might miss EXAF, and so with EXAF, when you go at the calcium NH, people show like you have some different short range order. Okay, I don't know. This is what they show. And for some systems, not for all, because actually in the URCHIN slide, she showed that the short range order of the ACC that she found in the URCHIN speaker. Sorry, it was the same as the one of CALCITE. But in some of the systems, some people argue that with EXAF you could see different short range according to the ACC. But again, it's element specific, so maybe it's actually more sensitive to PDF, because PDF we are average. So in the one peak you have average of calcium, oxygen, for example, oxygen ions. So what could be done? It's like some stimulation. So you can start to deconvolute and then do some stimulation and synthexes, but we didn't went to so far. But Capucine Sassoy, for those who know her, she's very good at that. So maybe we'll go over and try this. But I'm still desperately looking for some samples which will give some difference. We also tried the PURA, so it's a biogenic ACC.
Maybe a follow-up question. So on those samples, you don't have the exacts.
No.
Okay.
Nice, yeah.
Yeah, I mean, because if you see something different, that is,
Yeah, it would be the direct proof, exactly.
Then,
Yeah.
because
No.
so I had a similar question, like basically this disagreement, but maybe this is not disagreement, but this disagreement between the exacts and the PDF. What do you think about it? I mean, where it could come from? But okay, you answered, I guess.
Yeah, I mean, it's not because we don't see it that it's not there.
Okay.
Yes, everyone would argue always.
And related to that, as you showed the heating curves and the difference in the heating, did you monitor or review the water? It's really changing, not changing at all. The H2 over
Heating?
Yeah, the water, the of the face.
Oh no, well, of course, when you heat, like you start to artificially remove your water.
Yeah, but so you have the water removal.
Yes.
But it has no effect on the.
No, it does because so maybe I can. So when we start inducing the crystallization, there we see a bit of difference in between sample.
I was thinking when you try to differentiate
Here?
the ACC with pigment and the pigment, for instance.
No, by
you see
heating,
no difference.
no, with the pigment. No.
But did you monitor the water in the two curves? There is no evolution of water in the ACC.
While you heat?
Yeah.
Well, yes, when you.
Then why does it has no effect?
So it has an effect, but the fact that there is an aphthazine or not, it doesn't change so much. The water still goes the same way.
Yeah, but the effect of the water is really visible too. Because the PDF did not seem to change that much. Even I understand the differential between the PDF. There is no differential, but even the PDF seems to be very constant.
Between with and without magnesium?
Yeah, for instance, here.
Yeah, I mean, well,
how is it associated?
really bad. But
yeah, this one is fine. How is it associated to water loss?
Here, just because it fits with temperatures that we get from the DSC. Yeah.
But structurally, you understand the structural difference.
So okay, so what we propose is like, okay, so at the beginning we had that some water which goes away, and then the structural water goes away just when you have the crystallization. But all between, like it's not like happening at 200 degrees, it's like happening before, and all what is happening here that we can see like this changes slightly when you heat when the water is leaving, and when we add magnesium, here, like at the short, the medium range of all of 4, it's a bit different than here. Let me look at the paper. And what we propose is like the fact that magnesium is bound to water, which makes the water to get away differently. We don't have a full picture of it. But
my question was that in the case of the experiment with pigment.
Yes. There we don't.
You have that kind of residuals as well?
Yes.
Oh, okay.
Yeah, we do have this kind of residue. Yes,
okay.
yes.
Maybe I had two more questions. The first one is on so the sketch with the evolution of the trapping of some ACC within the spine. So I think you had a sketch. You had a drawing about. You had a drawing about how do you picture that. And so just,
it was really simple.
No, but I mean that's better. to understand. Yeah, I was wondering. I think it was after that, right?
Yeah. This one?
The sketch.
This sketch or the other one?
Maybe there was one later, right?
I like you.
Oh, yes, that's one. Okay. Original. In the beginning, so you have the ACC, you have some calcite, and you have the organics. You have crystallization of the ACC to calcite, and with the change of density, so basically you start creating some pores. And then you need their crystallization. It's more like
Okay,
sorry,
so,
this
no,
is
no.
the final step.
Yes, okay. So we're trying to, because we have little information, and one strategy is, as you said, like trying. So we have our. So basically, we burn. Well, we transform ACC, we burn the organic, and we end up with this. And now the whole point is trying to get back to what we had before. So you start to imagine, okay, this makes sense, and then. We try to understand as well, because here in this system, it's nice because we still have the ACC made biogenically, so we can induce the crystallization and try to understand, even if we induce by heating, which is not happening in the URCHIN, we try to understand then what happened in real life, let's say when ACC crystallized. So because of all this study, okay, what is happening now in the URCHIN when you have all the ACC? So ACC is crystallized, and at some point, but if you don't have organic, you can end up with avec Alberic, avec some big pores everywhere. So organics make it kind of filling the pores that are creating, and as soon as organics cannot fill these pores anymore, then it stops crystallizing. And we have this remnant ACC. Otherwise, you would end up with a crystal porous crystal. And when you look at the crystal at the lattice state, there is no pores. Well, there is 1 nanometer pore, 2 nanometers that we see with SAXS. This is yeah. I mean, this is our explanation.
And just another question on the related thing. So with the pigment, so I guess this is fluorescent, right?
Yes, it's not fluorescent.
It's also fluorescent. And so I was wondering, so are you trust on your sections, or I mean, are you removing them, the pigment for during the sample prep, or could you imagine, because that was one of the questions of the TEM,
yes,
where the pigments are,
exactly.
That's the structure. So is it still fluorescent, or
we didn't check yet.
Okay.
But this is something we were discussing. So now the sample, yeah, also they have been staying in those films, so we need to be sure that it's not affecting too much autofluorescence. But in principle, so in living cells with confocal, we see the autofluorescent signals. So why not? And it's supposed to be from the, well, it is from the pigment. So yeah, it's a good point.
Okay, so that's.
But we need the good resolution.
Yeah, and I guess the sample
with the
prep is
confocal,
a big thing.
You can go to that low.
I mean, you will never see that, but I mean, you can correlate, and at least you know.
Yeah.
For the signal will
Yes.
be detected. But then the big question is how the chemicals you add to that. That might,
we try to keep it
as native as possible. Okay.
Of course.
Yes. I know, but that's very interesting because it's hard questions that we have how to correlate those things.
We do also try over the CM with private fracture, but there you have no contrast, so it's very difficult to identify. And it's not one single cell. I'm not saying like the system is easy, but we have tons of different cells, and it's very difficult. So staining for us, it's at least a way to see where the membrane are and where they identify one cell. If you do just from the topography, when you do private fracture, sometimes it's very tricky.
Some cathodoluminescence in the electron microscope.
Yes, I'm not so familiar with the technique, but I heard about it.
That's you can apply to. But for TM, which is very, very tricky, but it would be fantastic. You can apply to this next time.
Yeah, well, we've been
for TM cathodoluminescence.
Yeah, we've been to Orsay to do some LPS stem hills, and there's this cathodoluminescence detector. Maybe we'll try something.
If the things are not gone away, it will be fantastic.
Thank you.
So I had one that was really fantastic what you showed about the connection between the cell, the red spheroid cell, and the mineral. And so you have a transfer of pigments. You mentioned there is a difference in chemistry. But when you think this, is there a selection by the mineral or something that is there before, or is there really a chemical transformation?
So we don't think it's a chemical transformation. I mean, this molecule, so this whole family of molecules, they can be oxidative. So they can be sensitive to one specific condition, but it has been very well. They know what people know in which they should transform. If like the molecule we have in the cells, one cannot become the other. So we have a mixture. So one is the most abundant, which is the echinochrome A. And when we have small others, and the echinochrome A is not the one which goes inside. So somehow during the pigment, it's released for some other reason. And it's known that actually echinochrome A is already part of the drug nowadays. So people are extracting like crazy to make some drugs. So, and in the case of the CHN, maybe it's the same. So this molecule is ready for the immune system defense. So CHN need it. So no way it's going to get into the spine. But the others that are just left over, and these are those ones which get into the spine. And why? I don't know if there is, because they are interact. So if we just look at the structure, so it's like it's this one which stays outside and this one which gets in. They're not so different. I mean, so I don't know if it would be chemically, because I would expect like they interact kind of similarly with calcium carbonate or.
One thing I was wondering in relation to that is that when you did this experiment of calcite growth, CSEO3 growth in presence of the pigments, are there some just complexation constants of calcium with the pigments? Are there just the basic calcium complex chemistry?
So yeah, they do complex. So in vascular, I did some experiments with so calcium ions with those pigments and NMR and see how much, and if I'm not wrong, one calcium complex to one molecule. So one calcium.
The two affinity of calcium for these two.
So they complex here mainly, like they do a complex.
Yeah, but are they very different, the affinity constants?
Between this molecule and this, it's just tried for an alpha-cycle. But it would be nice to try for the others. Yeah.
Simple model. If you have a difference.
Yeah, true.
And do you know if we can keep some remnants in fossils?
Some?
Remnants of these molecules or
So,
byproducts in fossils?
So in not in CHNs, but these molecules they are in other echinoderms. So echinoderms you have CHNs, little stars, starfish, crinoids, and
Cucumber.
I don't know the name of the scientific name. And in crinoids, in fossil crinoids, they found some naphthoquinones. They are not exactly the same. They are even more complex. I think it's two of them, but they identify some, and they are still very purple. It's a group in the journal.